Welcome to Nikolai's genetics lessons, and in this video I'm going to explain how to make a knockout mice. Why do we need to make such mice with broken genes? Imagine we have found 23,000 genes in human genome, for example, and we don't know function of most of them. So how we are going to find a function? We just make of course, we cannot do it with humans, so we using mice as model organism. Mice has almost all 23,000 genes which are homologous to humans. So we knock out such gene and we see what changed in organism of the mice, how phenotypically mice change, what effect such deletion of the gene have on the health of the mice. So later we can say that probably in human organism we are going to see the same result. So here's the problem. Your goal, you are studying how particular gene named ONO might play a role in panic attacks. You want to study mice that are missing this gene. To knock out the ONO gene, you will replace it with mutated copy that doesn't work. And in a few slides, I'm going to explain how to do it. Step number 1, you have to isolate embryonic stem cells that originated from male brown mice with normal or no gene, shown here in blue. Step number 2. In the second step, you have to add inactive gene with a marker. To the cells, add a copy containing a mutated inactive or no gene, shown here in red, and drug-resistant marker gene, shown here in a pink. Step number 3. Similar genes naturally swap. What does it mean? By mechanisms that are not completely understood yet, similar genes will swap places. The ONO gene plus drug resistance marker gene is incorporated into the genome and the normal version is knocked out. This process is called homologous recombination. Step number 4, you have to add a drug. Cells that doesn't incorporate the inactive ONO gene don't have the drug resistance marker gene, shown in a pink. Adding the drug kills cells without marker and leaving you with only cells that have an inactive version of the ONO gene. Step number 5. At this step, you have to grow chimeric mice. By transplanting stem cells that carry the inactive ONO gene into the white mice embryo, you'll create what is called chimera. Chimeras have patches of cells throughout their bodies that grew from the white mice cells and patches that grew from the brown stem cells. Some of the cells that have the inactive ONO gene may develop into reproductive cells. Chimeras are easily to identify because they have both brown and white patches of fur. Step number 6. Mate male chimera. If male chimera has some reproductive cells, which is sperm, that originated from the brown stem cells, he will produce some brown offspring when mate with a white female. Step number 7. Test and breed brown offspring. Half of the brown offspring will have a copy of the inactive ONO gene in all of their cells, including their reproductive cells. These mice have one normal copy of the ONO gene from their mother and one inactive copy from their father. So half of their reproductive cells will contain a normal copy and half will contain an inactive copy. These mice can be identified by performing DNA sequencing of their ONO genes and then breed with each other. Imagine that genotype of one mice is normal chromosome with normal gene on it and another chromosome with defective gene on it. And genotype of this mice is also one normal chromosome with normal gene and one chromosome with defective gene on it. Now let's build simple Punnett square. And let's say that one is going to be mother side, another one is going to be father side. So father side here, mother side is here. So we are going to have following genotypes. So both of them, both parents are going to be heterozygous. So two normal genes we expect here, one normal, one defective knockout here, one normal and one defective inherited from the father side here, but here we are going to expect two defective genes, two chromosomes with defective genes. So 1/4 of the progeny are going to be knockout mice. Congratulations, you have made knockout mouse. 1/4 of your resulting offspring will have two copies of the knocked out or inactive or no gene. You can now study these mice to determine how lacking a no gene may affect panic attacks. Subscribe and see you in the next video. Goodbye.